Good morning, everyone. Can everyone hear my voice?
Yes, Mengti.
All right, that's good. So, good morning, everyone, again, and welcome to today's webinar, Resolve Complexities in Cancer Research with Spatial Transcriptomics. Thank you all for joining us. I'm Meng T, and I'm the moderator for today's webinar. Before we begin, a quick note on this housekeeping. This session will run for about 30 minutes, and we will have a Q&A session towards the end. If you have any questions during the webinar, please feel free to type them in the chat box and we will address them after the presentations. To ensure the best experience for all the participants, please keep your microphone muted throughout the session. In today's webinar, we are delighted to welcome Mr. Chi Hong as our speaker. He is the Field Application Manager at NextGene Scientific, bringing him over a decade of experience in the field. Today, he will share valuable insights on 10x genomics and explain how spatial transcriptomics is helping to address the complexities of cancer research. Without further ado, let's get started. Ji Hong, the floor is yours now.
Okay, thank you, everyone. I hope that my voice is clear enough. So I guess recently there is a lot of flu. It's a flu season, so I think I caught some flu as well. So my voice will be a bit rough. So I hope that it doesn't affect your experience of listening to today's webinar. So I'll be sharing my slide today. Okay, one moment. Minty, can you see my screen? Minty?
Yes, we can see.
Okay, thank you. So first of all, very good morning to everyone who use their time and spend their time to join the webinar today. So my name is Chi Hong and I'm from NextGene Scientific. Today, I would like to be your tour guide to bring you on a short journey on the exploration of the spatial transcriptomic world. So I would like to bring you to venture to this world to try to see what spatial transcriptomic can offer and do and potentially help in your research and potentially as well to assist to answer your research question. Okay, so the webinar today will be mainly focusing on using spatial transcriptomics to try to resolve some of the complexity in cancer research. Okay. So the technology that offered by NextGene to help and to perform the analysis of these spatial transcriptomics is by 10x Genomics. So there are two platforms that 10x Genomics offer to perform these spatial technologies. One of them will be the Visium Spatial, and the second one will be the Xenium Insight 2. Okay. So the only difference between these two is that the Visium Spatial Transcriptomic will be investigate the entire whole transcriptome of cell, and it's actually sequencing based. Whereas for the Xenium, it's a system that is more on targeted genes. So we zoom into certain set of genes, and then we see, then on that we investigate the expression of this targeted gene in the cell that we are investigating. Okay, so before I venture into the world of spatial transcriptomics, so I would like to bring everyone into a scenario following this. Okay, so let's say we have go into a clinical setting in the hospital. So we have a very anxious person because he has been informed by the doctor that there is some abnormal growth or lumps in a part of his body. Okay, so advised by the doctor that a further investigation is required to look at this particular abnormal growth. Okay, so one of the suggestions is that they will proceed with a tissue biopsy. So let's say this particular procedure has been performed and the tissue biopsy has been acquired. Okay, so it has been prepared to become a FFPE block. Okay, basically they treat the tissue and then with some formalin fix, and then after that they make it into a block, and then we slice this block to make it into a tissue section. Okay, so this is the normal procedure because we want to look at the tissue morphology and the cell of the biopsy. Okay, so from there we are able to stain the section, the FFP section, using H&E stain, and then we look. I mean, not we, but a very experienced pathologist will be actually investigating the tissue slide to look for the sign of, let's say, whether the growth is actually benign or actually is aggressive tumor. Okay, so from the tissue slide, there will be surrounding by normal cell as well, and there could be some lesion that showing sign of, let's say, hyperplasia or even, let's say, tumor growth. So depending on the grading of the stage of this abnormal growth, okay, so it could be invasive and potentially metastasis to other site. Okay, so based on the experience of these pathologists, they will actually grade it to be a benign growth that actually is more like harmless tissue biopsy, or actually is a potentially a different grade of cancer. Okay, so to further assist this particular investigation, so sometimes in certain type of cancer, we'll use the biomarker, basically an antibody that actually will bind to a specific protein on the cell. This protein actually has been confirmed to show the sign or the information that whether the tissue belongs to a certain type of cancer or not. So the particular procedure that can use is actually called immunohistochemistry. Okay, and then we will actually have a better view of the condition of the cell. Okay, so going forward from here, that will be more or less about it. So based on the experience of pathologists and also the tissue morphology, that will be the diagnosis in the clinical setting. So the thing is that, is this information enough? Okay. Because we know that tissue are very complex, there are many different types of cell over here, and whether with the information of the cell morphology, so we are able to differentiate it and separate them properly, and also whether the different type of cancer cell, we're able to separate them correctly or not. Okay, even though with the help of the biomarker, sometimes there could be only one or two biomarker, means protein that we have and access to actually differentiate them. So will that be enough? So over here in spatial transcriptomics, we are offering a deep dive into the cell itself. So we zoom into the tissue and then to individual cell, and then we get the entire transcriptome of the transcript of this particular cell. Okay, so we get all these cells. Okay, let's say there are millions of cells over here. We get each individual cell gene expression profile, and then we actually separate them based on similar expression genes. Okay, and then we categorize them into different type of cell based on those expressions. So will it be a better useful information that further assists that for the pathology or clinical setting to look into what type of cell we're actually looking at, and what is the progression and what is the prognosis of the tumor progression. Okay. So here I would like to show you how complex in tumor setting. Okay, so we have a normal cell like smooth muscle cell, we have a normal endothelial cell. Okay, then we have the epithelial cell. So there could be more than one type of epithelial cell because we have the normal one as well as the tumor cell. Okay. Not to mention that we have some immune cell as well on the side, because when we have tumor progression, the immune cell will be going into the tumor site to try to fight against the cancer. And therefore, we will actually at the same time detect some immune cell. So therefore, with over here, we can see that there are at least 3 different types of cells, and not to mention that there are many further differentiation into the type of cell that we are showing here. So it's indeed a very complex situation. So we might not be able to differentiate them very well by using just the histology site morphology dependent. Okay, so just now we are showing how complex could be a tissue slide that actually consists of many different types of cells. Now, if we actually zoom into the tumor itself, we all know that tumor are actually highly mutagenic. Okay, so they keep on mutate, and then after that, how to say, they follow the Darwin's rule where survival the fittest. Okay, so once they manage to mutate into a certain type of cell that rapidly grows, it will actually outgrow the rest of the cell. Therefore, even though we are talking about tumor as a whole, it is actually consists of many different type of cell. So the strongest one, the most aggressive one, will be in the end outcompete the rest of them. Okay. So therefore, even though we grade them as a single type of tumor, inside within this tumor cell, there could be a different cluster or category of cancer cell that acquire different chromosome aberration. Okay, and this particular mixture of tumor cell can lead to treatment-resistant clone. Okay, because the one that actually grow aggressively and potentially have resistance over the treatment will be eventually become the dominant cell. Okay, so therefore, when we look at the histology side, we might not get this information. But if we actually zoom into the gene expression level, we might actually get additional information. So we actually combine these two information. Okay, so therefore, what is spatial transcriptomics able to offer is that we are not only looking at the histology image, okay, but we are actually at the same time looking at the gene expression level of the individual cell at the same time. So we are combining the information from the histology image, let's say the morphology of cell and how they actually organize. So whether there are different area of tumor cell and also what kind of cell is actually surrounding like fibroblasts, normal epithelia, okay? And then we actually combine with the gene expression level. So after acquire this particular information, we actually overlay both of them. So we are able to see not only the morphology of cell, but the expression pattern. Okay. So after we acquire this information, we can actually separate the cell properly. Okay, with added information from the gene expression level. Okay. So here I would like to show how actually we're able to get the transcript information of the individual cell. Okay, so this will be the what we call a capture slide. So on the capture slide, we have the capture area. So on the capture area, we have all the oligo. They are able to actually bind to the RNA transcript. So when we actually overlay the tissue to the capture area, okay, we permeabilize the tissue to allow the RNA to move from this dot to this particular area of the capture area. Okay, so the capture area will have a spot that actually have unique location barcode. Okay, so after we actually sequence all this RNA transcript, we actually map it back to the location based on the unique barcode. Okay, then we combine them together to get the information of the cell morphology as well as the gene expression. Okay, so here will be an overview of the workflow. So basically, we have a tissue biopsy, then we process it either using FFPE or fresh frozen method. Okay, so after we get the tissue block, we actually section the tissue biopsy into a slide. Okay, we put it into a slide, and after that, we go through the entire workflow to transfer the RNA transcript from the tissue, I mean the cell, to the capture area, and then we sequence it. Okay, and so after we get the sequencing result, all the RNA transcript will be actually mapped back to the area of the tissue site. And then we get the information of all the gene being expressed on different region of the individual cell on the tissue. Okay, so this is one of the example able to show you. So this will be the tissue. Okay, so we have different color representing different type of cell. So we say that is a different type of cell because this cell or area, the cell of this area has a similar gene expression profile. So therefore, we actually cluster them, group them together. So then we have different area, and then we actually use different color to represent different type of cell. And over here we actually call it cluster, but we can rename the cluster into a cell type. Okay, so at the same time, we actually can able to see the gene expression information of different region of the tissue as well. Okay, so how can we actually apply spatial transcriptomics into cancer research? Okay, so as mentioned earlier, so we get the gene expression information of different cell type, and then after that, we actually map back to the location. Okay, so we get the gene expression of all the different cells, and then we actually identify the cell type. Is it a normal cell? Is it a biofibroblast, endothelial cell, or actually is actually a tumor cell? Okay, then we see where are the tumor area on the tissue side, and then we look at the gene expression profile of those tumor tissue. Okay, so this is another example. We have the histology image information of cell morphology, and then we combine with the gene expression information from the spatial transcriptome. Okay, so one of the way that after we characterize the cell type, we actually can zoom into the specific gene as well. So over here, we are showing that area where there are high expression of this ependymal cell marker, CCD153. So this particular red color, I hope that you all can actually see it as well, and this area. So these are the region with high expression of this CCDC153. And for this another gene marker, CLIC6, so you can see that it's highly expressed on this particular region as well as here. Okay. So this is, we can actually investigate the cell type, or in another way, the reversely, we can actually investigate the target gene that we're interested as to perform our analysis. Okay, so this is an example that after we characterize the cell on the tissue side. So over here we have a myoepithelial cell that actually has this particular marker, ACTA. And then we have another type of epithelial cell. We have the ductal carcinoma inside the non-invasive carcinoma cell. And we have the invasive tumor. So all of these have different color. So the invasive tumor is actually labeled as red color over here. So in the end, if you look at the combined with the morphology, histology information, you can actually look at the area of invasive, the non-invasive ductal carcinoma. That actually there are two types over here. Okay, and then we see how we can actually use this particular information. Okay. So is that all that spatial transcriptomics is able to offer? So basically cell type, and also let's say gene expression, and lastly marker gene. So why this information are important? So over here is actually showing an example how we can use this information to look at the prognosis of the treatment. Okay, so the region that we actually highlighted in yellow here will be the tumor area, the tumor cell. So the same over here. Okay, and then this is after being applied with a treatment. Then we actually see whether the treatment cell, which is actually the T cell, how they actually are able to infiltrate the area. So this is the hot tumor with good prognosis because the T cell that being used as a therapy is able to cover the entire area of the tumor. So the doctor can actually predict a good prognosis because this T cell will be able to use to target the tumor cell and potentially curing them. Okay. Well, in this particular treatment, we see that this T cell is only cover a small part, and borderline of the tumor is not able to go into the tumor, the core. Okay, so therefore you will not be able to kill the tumor cell, and therefore it's actually having a poor prognosis even though with the therapy. Okay, so now I would like to show you what some of the example of what spatial transcriptomics can offer in terms of information after the running of this particular experiment. Okay, so spatial transcriptomics in this particular experiment is which is actually using a colon cancer tissue, which is actually a FFP blocks. Okay, we are able to offer a high amount of information. They give you high resolution information on the location. So there are 18,000 genes detected in this particular spatial transcriptomics, and we can actually get very, very precise information due to the genes that we are able to detect. Okay, then we can actually relate the information of the gene versus the tissue morphology. Okay, so with the latest improvement from our free software, we are able not only to offer gene expression information, but at the same time, we try to draw the border of a single cell. Okay, so we are able to draw the border of single cell, and then after that, we look at the comparison of between different cells and how they respond to each other. Okay, so next we'll move to the next section of the day, which is actually looking at spatial transcriptomics on tumor heterogeneity, biomarker, and immune cell infiltration. And then later on, we'll look at some case study that how the different group of researchers using the spatial transcriptomics for their research interests. Okay, so here I would like to show you that spatial transcriptomics is able to reveal a different type of cell in a tumor. So this is actually a human lung cancer tissues section. So using the spatial transcriptomic information, we are able to identify the epithelial cell, okay? But we are able to differentiate the different type of epithelial cell. So based on the data produced by spatial transcriptomics, we have two different type of epithelial cell here. So we cluster them into the purple color here, the cluster 1, and then the green color into cluster 2. So although they are both epithelial cell, but they have a different gene expression profile. And at the same time, we can actually look at the marker genes being expressed, specific marker being expressed in cluster 1, let's say, versus the cluster 2. Okay. Next up, this is actually again showing the different type of cell they are able to detect using the spatial transcriptomics. So the example here is actually human breast cancer. Okay, again, we identified the cancer epithelial cell, and then together with the help of the tissue morphology H&E stain. Okay, so we reliably differentiate known cancer marker expression under the tissue section. So over here is ERB B2, so blue color. Okay, and then SLPI, and then CYP B1. Okay, so here I'd like to show that in ductal carcinoma in situ tissue section. So again, we are able to have the H&E staining information. We overlay with the gene expression information to identify the cell type. So the red color over here is actually the breast granular cell, and then myeloepithelial cell, and even the immune cell. Okay, then after we combine the image here into this particular information. So we get the both type of information using the spatial transcriptomics. Okay, so this one not only we get the cell type, we further look at what kind of unique biomarker or genes expressed on certain part of the tissue. So let's say the granular side have a unique marker gene as ZGP1, and this is to characterize the cell type using the marker genes. And also, of course, the breast cancer marker. So we actually see what are the distribution of this breast cancer marker on the tissue cell. Okay. Another important research interest on cancer will be how the T cell responds to the tumor. So do we see a lot of infiltration of, let's say, T cell, B cell, and natural killer cell? And where are they? Okay, are they overlapping with the cancer region, or are they actually on the border of it? Okay, and then not only we are looking at the location and whereabouts of all these immune cells, but we can actually see the gene expression profile of the cell. So we know that, let's say the useful cell will be the cytotoxic T cell. So we will see whether the T cell actually expressing those cytotoxic or even cytokines to help to actually fight against the tumor. Okay, or the tumor is actually able to suppress the function of the T cell, and so that making them not able to fight against the tumor. Okay, so those are the information we can get by looking at the immune cell and also their gene expression profile. Okay, so next I would like to share a few case study on spatial transcriptomics to be used on cancer study. So in this particular publication, it's actually a nasopharyngeal cancer, which is actually very common in our region. Okay, so in this particular publication on NPC, it showed that the data generated from spatial transcriptomics show that there are enhanced development and suppresses activity of the T regulator cell through the interaction of two markers, receptor and ligand. Okay, so the cancer cell actually express this particular marker called CD70. And then after that, they're actually using it to interact with the CD21 of the T cell. Okay? So to actually promote the activity of the T regulatory cell. Okay, because the T regulatory cell is actually immunosuppressive cell that will actually causing the reduced activity of the cytotoxic T cell to fight against the cancer. So therefore, when we overlapping the location of the NPC as well as the Treg, okay, so we found that they are highly expressed of a CD70, and the patient sample that we use over here are those that have a poor prognosis. That means that the treatment actually not really doing well. So then they actually come into this, in this publication, it come into a suggestion and conclusion that this CD70 expression of the NPC actually causing the Tregs to actually infiltrate all this region and suppressing the activity of the CD8 T cell. Okay, so here is another publication using the spatial transcriptomics platform Xenium to identify immune and spatial biomarker of therapeutic response and resistance. Okay, so here the tissue is actually as a result of after treatment of drugs, and then after that the information are able to provide insight into the cell level interaction. So between the cancer cell and the cytotoxic T cell. So from the data, it's able to reveal that there are significant correlation with the treatment response. Okay, so the finding found that the cytotoxic T cell are correlate with better response when they are HR negative cancer. Okay, if there are a lot of HER2 strong positive cell, you actually with poorer response of this particular drugs. Okay, again, this is another application of spatial transcriptomics to look at the drug response, which is actually the neoadjuvant, okay, and there is anti-HER2 antibody therapy. Okay. So in this particular publication, it actually investigate the effect of this particular drug, and they are using a combination of the computational the prediction as well as the data provided, generated by the Xenium platform. Okay, so the spatial transcriptomic platform Xenium is used to target focus on 380 genes. So the data generated is providing precise cell type identification and annotation of representative gene marker. Okay, so by using this information, we identified cancer, immune, and stroma cell, and they, based on the biomarker information, they classified the cancer cell into whether they are HER2 strong, weak, or non-responsive to the therapy. Okay, so this is another publication. Okay, so understanding the resistance mechanism in glioblastoma treatment using the spatial transcriptomics. Okay, so when the experiment started, it's actually using a drug called BLZ945, which actually inhibit this CSF1R receptor. Okay, so the spatial data is show that it will improve tumor regression and survival in the mouse model. Okay, so however, in some cases, the application of this particular drug alone will trigger some response from the glioblastoma, and actually promote tumor survival and recurrence. So under this condition, this group of scientists actually use a combination of drug treatment to see what's the effect. So therefore, they're using combination of BLZ945 and 2 other reducing agents to enhance the effect of the therapy. Okay, so how spatial transcriptomics help in this particular understanding of the interaction of the drug treatment and also the effect. It actually is able to provide information of gene expression and cell interaction in glioblastoma, revealing how certain type of niche cell is able to form and protect the dormant tumor cell. Okay, so this is crucial for identify when and where to intervene for better treatment outcome. Okay, so that will be my last sharing on the case study. So as a summary of today's journey into this spatial transcriptomics, so the first one will be the spatial transcriptomics offer gene expression of individual cell to offer more information on cell type and cell state, in addition to the histology, cell morphology information. So the high resolution information offered by spatial transcriptomics are able to give information on tissue organization, how they arrange into a tissue, and the morphology information, cell type characterization, and at the same time give us the marker genes location, functional state of cell, referring to the immune cell. So whether they are actually being able to carry out their job, and how the immune cell interact with, let's say, the tumor cell. So in short, spatial transcriptomics is a promising tool to provide insights into tumor heterogeneity and biomarker discovery and immune cell interaction to tumor. Okay, so with that, I will end my sharing on today. I hope that everyone get some useful information and able to potentially use the spatial transcriptomics to answer some of your research questions. Thank you.
All right. Thank you, Chi Hong, for the insightful presentation on cancer research using spatial transcriptomics. Before we begin the Q&A session of today's webinar, we would like to announce an exciting program, the Design Your Dream Experiment with Single-Cell and Spatial Transcriptomics program. Through this initiative, researchers can apply to receive exclusive discount and technical support for their single-cell and spatial transcriptomics research with 10x Genomics. So you can scan the QR code on the screen or click the link in the chat box to apply. For more information about the program, please do not hesitate to contact our friendly product specialist. And now we will move into our Q&A session. You can unmute yourself to ask a question or feel free to type it in the chat box. Yeah, do we have any question from the floor? Well, we have one question from Akila. Does spatial transcriptomics works for microRNA studies?
Okay, thank you for your question. So for now, we are unable to sequence the small RNA, the microRNA on the tissue because due to the arrangement of the microRNA because they are too short and they don't have poly A. So therefore, we are unable to recover the information of this microRNA. However, if let's say microRNA is being used as a applied to the, you inject and treat the cell with the microRNA, okay, and you transfect them and to see the effect of the microRNA, how they actually regulate your tissue and all that. Okay, so then it's possible.
All right. Do we have any more questions from the participants? We have another question from Fong Fang Chen. May I know, in view of its RNA degradation in FFPE samples, do you have a cut-off time for sample freshness? How reliable it is compared to the use of fresh frozen samples?
Okay, thank you for your question. I think that this is a very a good question that I actually didn't cover because we all know that FFP sample actually it has degradation problem, especially those storage for a long time, and so therefore in 10x Genomics we actually have a method to actually to deal with this particular degradation. So as long as the depending on the technology that we are talking about, we can actually accept the RNA degraded not more. That are not shorter than 200 base pair in length. Okay, so the technology that we use is actually we use a specially designed RNA. Probe to bind to the set of predicted transcript. So basically, for now, this technology we only offer for the human and also the mouse sample, where actually the probe is designed to cover all the entire transcriptome of the human and also the mouse. Okay, so we design the specific probe to bind to those known genes, and then we look at the gene expression. With that, we are because the probe is actually shorter, so therefore they are able to bind to the degraded RNA. Okay. However, if let's say we are using let's say a fresh frozen sample or FFPE sample that is not human, that doesn't have all this design of the probes, then we will actually need better RNA integrity from the FFP sample. So therefore, before we actually proceed into this experiment, we need to section the tissue from the FFP sample slide. Then after that, we actually extract RNA from there. We will actually see what's the quality of the RNA in the sample. So if we proceed with the probe method, then we actually can accept a more severely degraded RNA condition from the tissue section. But however, if we are using, luckily today we're talking about human tumor, so therefore it's not an issue because it doesn't matter it's a human or mouse FFPE, we can actually accept pretty low quality RNA from the FFPE sample. So I hope that answers your question. Thank you.
Chi Hong, we have another question from Yong Gong Yi. I would like to know if I have tumor tissue fixed in 10% NBF, but not yet processed into FFPE block. Will overfixing the tissue, for example 7 days, affect the spatial transcriptomics workflow? What is the optimum fixation time recommended?
Okay, thank you for your question. Actually, in 10x Genomics, in order to have the good result from to generate a good data from and also good result from the FFPE sample, we actually has some recommendation on how we actually fix the tissue and also the workflow to process the tissue biopsy and all that. So we have a sort of a guideline PDF from our 10x Genomics site that actually we have an entire procedure how we should process the sample. So that will actually give us the best result. Okay, so if let's say there are some deviation from the workflow, then we might actually need to see whether how it will actually impact the result from the spatial transcriptomic data. Okay, so we need to evaluate them first. So therefore, if you're interested, then we can actually connect with us. Then we actually connect you with the expert from Therapeutics Genomics to see whether your workflow is actually acceptable for the spatial transcriptome. Thank you.
Thank you. Is there any more question from the floor? Yeah, it seems like there is no more question. So now, thank you again, Chi Hong, for the informative and engaging presentations today. And also a big thank you to you all for participating in this webinar. We hope you find the information shared here is valuable and inspires you to apply it in your own research in future. Before we end the webinar today, we would love to hear your thoughts. Please take a moment to fill up the survey form in the chat box. Also, if you have any questions, feel free to contact us at sales@nextgene.my. Thank you once again, and we look forward to connecting with you guys again in the future.
Hey, is it a raised hand
I think
or accidental raised?
I think they should be accidentally
Okay.
pressed the. All right, thank you. Bye bye.
Bye, everyone. Thanks.